today is a molecular biology section. And what is molecular biology? You know, I will start at molecular. Molecular is study about chemistry interaction between a molecule. Molecule is a you will say it?
Two atoms combine to each other?
Okay, which one? Okay, let's go
Two
first.
elements combine to each other?
Yes, correct. Yes, a molecule is a group of two or more atoms that held together by a chemical bond. And biology is study about life and organisms, including structure, function, growth, origin, evolution, or distribution in life. And molecular biology is a combination of these two fields to study about molecular basis of biological activity, including synthesis, modification, mechanism, interaction, structure, and composition in cells.
Basically, if you understand in the concept here, it's like you understand about the about building block, right? Which is like the molecules, right? Combined from one atom to two atoms. But this is more complicated, most complex. So we have a lot of molecules inside. Everything is it's been composed of molecules. But now we want to understand how the molecules interact with each other and what the meaning of like they interact and give something like a function, something like that. So like inside the cells as a unit of living cells. But think about it, you have many cells put together to be a tissue, what function, stuff like that, right? This is something beyond that, right? But actually based on the fundamental of molecular biology, that's what we try to present to you to understand from the chemical structure until they be complex and we can communicate things like that and make sense of life. Okay? You see that Pian want to present on that. This is like talk, like small molecules here, so the atoms making a molecule. Okay, not molecule right there, okay? Now it's a molecule to be a more complex, which is synthesized to be like a little larger, from small molecule to be larger molecule to be called macromolecule. And then inside there, some of this would be like inside, like now in the cells, really small in the cell here. Inside there's like organelle, like the function is this for example like this picture. What is it? Picture. You know what the picture is?
The diagram?
What's the picture is when it says like organelle? What kind of organelle here? Inside the cell.
What is this?
What is it? What is it? What is it? This one. Okay. Now this is cell, right? This is cell. It's a cell, but there's little tiny teeny things. It's like a part of the machines. It's a part of the machines. And this picture is one of inside the machine. We call the organelles, like organelle, right? The picture like here, the picture, what they call? What's this called inside the cell?
Mitochondria.
Mitochondria. You know what's this for? What's the function? What is function? Energy. Here we go. Give us an applause out here. Very good. So the energy inside the cell we call it ATP. We'll talk about it later, okay? But that is one of the things that is like fuel. What's we call ATP. Alright? Okay? I didn't try phosphates, for example. It's inside there, so there's like organelles, to keep things like functions as cells. Alright? In other words, it's cells, they combine together to be like a tissue types. So they function as a certain things. For example here, like let's see, like kidney, right? Kidney, they kind of, take care of almost everything in the body is like detoxification. You know detoxification? You know what I mean by detoxification?
Get rid of the bad stuff.
Exactly, yes. That's one other thing, okay? And okay, that's the thing. And also, what about the heart thing?
Pump blood around the
Pump
body.
the blood around. It functions as a specific certain functions. Okay, the heart's pumping things like that. Okay, the liver does things like that. But actually, they function exactly the same cells, same genome. They have the same genome. They are the same genome. You know what a genome is?
genome.
Genome, yeah, when I say genome, we have the cells, we have a nucleus. A nucleus, inside the nucleus contains,
Are there genes inside?
what's that?
Are there genes inside?
There's a gene inside. Okay, the gene is a piece of like a DNA that put all together to be a certain information that can do something else. But the gene, many genes, they connect each other as a genome, the whole genome. So many genes inside there. Okay, we call the whole thing is a whole genome. Okay, thanks a lot. Okay, okay, understand. All right, but each of tissue, if a single cells here, okay, almost all of them, they have the same genome, but they function differently. You get my point now? Okay, that's what I was just simple explain to you what is inside genome here. But remind yourself, we talk about here is molecular biology. Okay, all right. Okay, now I just talked about this organ, from tissue to the organ, so the whole system and the organism to be keep us survive and function normally just like that, right? So what we want to say here, understanding about the cells, how they function, about understanding about the genome, is important to keep us survive, to keep us function normally. If something gone wrong, can we fix it? Can we treat it? Can we prevent something? You see what I mean now? This is important. Why we have to learn about the basic understanding about molecular biology, particularly for DNA, which is going to be our station today. All right, give you station.
And I will talk about DNA. DNA is a molecule that carry genetic information for development and functioning in organisms. DNA is a double helix form and the shape look like a star because it is pairing by hydrogen bond. In DNA, it have 4 nuclear bases, including A, T, C, and G. A is adenine, T is thymine, C is cytosine, and G is guanine. And you see a picture, A will pair up with T, and C will pair up with G by hydrogen bond.
This is from nucleus, right? Okay, from nucleus. Now, we go to like a bit on the genome. And the genome, we talk about what? A gene. Okay? Actually, this could be a part of the gene, okay? This would be a part of the gene, okay? From a larger to macro size, this is smaller, deep down to that. Now, see, it's a piece of DNA. It's a piece of DNA. We just like cut just one part of DNA, which is a part of the genome, the whole genome, the whole thing that contains all the information right there. When they say information is like this. This is information. It's just like a code. When they have like A, T, G, C, C, this is a code. There's a meaning. When you say we write the code, right? There's a meaning, different meaning, okay? And the meaning is important. What is the functions of the one? But that is a code. It's a code contained inside the DNA as a gene. You get it? And that's a code right there. Okay? But the code is important because the code can be arranged differently. But there's a meaning. There's a meaning. Okay? There's this the cells, our part, the ribosomes can be just like that. Okay? All right. Once we have that understand, the building block of DNA is the pairing, okay, of the what they call DNA. DNA is a building block. So DNA is a building block. Okay. So we have like DNA, G, we call it G. We pair with C. Okay. C, G depends on. Okay. We'll talk about later on. T with A. Okay. And the bonding is a hydrogen bonding. You see that? This interaction right now, see that? Remind yourselves about how can they interact, how they build first, how they interact, how they contain as a DNA structure. You get it? Okay. So after that, we go to like a little bit closer look inside what does a molecule look like to you then? Okay. So when they say DNA, actually it's polymerization of like For example, like this one, guanine structure like that. This is a base pair guanine, okay, with the sugar base, okay, and the link to the other DNA common as a C, which is what cytosine, okay, link to that what was ester bond right there. Will be what A, which is an adenine, for example, right there, okay, and then in anti-pair each other, when pairing, to be like a stack like that. Okay, you follow? You follow? This is what the DNA structure look like to you. Okay, which is like you will be extract out today from the
Fruit.
it's a fruit. Okay. Alright, okay, go ahead. Basically here that is where to be extract the DNA out. Can I ask you again? Where is the DNA? Inside the cells.
The nucleus.
In the nucleus, right? Okay. So in order to get out of that, what's your thought about it? How can we get it out? How? So they need to be like no, just chemical, just like that. This is how. How to get the DNA out? Good.
Or use alcohol.
Don't need to worry about alcohol things here. I'm just like get the idea how to get the DNA out. What's that? Break the nucleus, right? That's the ultimate goal right there. Okay, give her the applause right now. I need to like okay, listen to that. Okay, so break the nucleus out, right? Because the genome, right, is inside what? The nucleus, right? But before that, you got to break what first?
Nucleus.
No, you got to break what first?
Being
Cell.
cell.
The cell. Thanks a lot. Thanks. Okay. You guys great, man. All right. You got to break the cell first. It's like either the cell membrane or the cell wall. Depends on what?
If it's a fat or an animal.
Exactly. That's correct. That's correct. So you got to understand. Okay, how to break the cell wall? How to break the cell membrane? It depends on what kind of reagent, right? What kind of solution you're going to break on that. You got to understand where the tissue comes. You see my point here now, right? Oh, go back to that. Okay, could go back to the tissues and stuff like that. That's easy. When you got a single cell, that's easy. You got to break what? The cell membrane first, right? And do what? Break the nucleus. Get the genome. Okay. But eat this one. You see that? How can we do so? So it means what? Okay. My question is, if we got a piece of tissue here, how would we break it in and out? What kind of process would that be first?
Taking one out. Taking
How?
one cell.
By hand.
Like dissolve.
Dissolve it? Okay. Okay. Okay, keep you guys are great, man. You got to get the single cells out, right? Now, think about the biology here. When the cell, with the tissue, they have many cells, right? And then they link with some other like proteins, whatever, okay? For example, to link like acid glue. So we got to wash the glue out, right? So in our like biology world, we got to use enzyme to clean off the protein that link the bridge the tissue all together right there. You understand it now? Okay, that's how you picture things here. How to get squeeze out the DNA, but you got to understand where the sample from first. Okay, see that? Now assume here we got a single cell. We have a single cell for sure. Let's say we process everything here. We get a single cells out. We add we call the lysis buffer, to break down the cell membrane, and also maybe break the nuclear membrane also at the same time. So we got lysates, so mixed moves, everything's like all together right there. Okay, the DNA, the part of tissue, stuff like that, or cell membrane, everything's like all inside there. Okay, once it's finished, okay, that's you kind of pass through some column. We have a membrane over here, all right. A DNA will stick to it, okay, because they're the way to stick to it, okay. Like you keep that the column with the DNA inside, okay. We put it to the other one. You washed it out, okay. So then you will wash through that, okay. Elute it, washed it, and elute it. You wash it first because then it's stuck right here. You wash it to clean up all the thing here, and then you elute it out. Okay, there's like a procedure to do so. So that basically you understand today about how to get the DNA out. Okay.
I have 4 examples to show you what built-in is for. At first is DNA profiling. DNA profiling is a technique for identifying individuals or sample by DNA profile. So DNA profiling is used in forensics to identify a criminal. And this slide, I will show you.
Have you seen it before? Have you seen it before? No? Okay, now I'm just seeing that's the real thing. You want to make sure that this is my dad or not? You can do so. You can do so. You can do so right now.
Yes, you can do it. This is called a paternity test. It's a test for search who are paternity of a child. And for example, who do you think is a father of a child? A, B, C, or D?
B. B.
D. B. Oh, okay, is a correct answer. B is a father of a child.
Okay.
One, yes. 1, 2.
Okay. Before I explain a little bit more, every single person, they have a unique DNA sequence. Okay. Basically, we have the same genes for sure, but somehow the DNA, the difference in terms of sequences. Okay, that's unique for individual is important, and that is how we can design it to check whether that what part of your DNA is, and this your profile, and this is your profile. Okay, each individual they have different, they're all different. Unless what?
Sibling. Or
Sibling is not the answer. True. Unless what? Unless we are
family.
twins.
Oh.
That's correct. We're twins. We're twins. Correct. Thanks a lot. Good. Very good. Sibling is just like individual person in terms of like a genome sequencing, okay? But twins, they're identical. But behavior, it's not the same. Okay, we just talk about something which is not the same, but the same is a genome, okay? Among twins, all right? And this is how we decided, okay? That's a way we call like, like check the uniqueness in terms of profiling, so we can see whether that, all right? Okay, cool. Are they then? Who's the parents? Yes, I want to check my parents too, actually. Whether that is it my real parents or not? Okay. All right, mate.
If we want to test our parents, do we have to test us and our parents too?
What do you think?
Yes.
What do you think? Supposed to be yes then. Because like, okay, why your parents, your mom, your dad sleep. And just pick up some like what? Maybe it's like hair, whatever. Just get there and I want to test myself. Then all right, you can do so. Okay, go ahead.
And we use DNA for medical tests.
I just go back again. So what else I can use for profiling techniques to other applications other than just to see that this is my parents or not? Any application that you can think of about this kind of DNA profiling?
The face looks the same.
What?
The characteristic.
No. I'm asking right now is the after you understand about this one application, right? We call DNA profiling. One of the things we can check your parents that we what do you call it in the word? Parental identification, right? Parental identification. If any other application that you can think of based on these techniques, okay?
Crime scene and this.
Crime scenes and true. That's so true. You see that when they get the blood with the little shams, go to the crime scene first. You get the blood sample now, and the people exactly the same thing in terms of the data extraction. Exactly the same thing, the same procedure right there, right? And afterward, you check that, and all right, I got the same profile here, right? It might be suspect. I ask you to come to the police station here, and get your blood out, and I want to check on it. Right? If the data profile is not the same, like could be this guy, okay? It's going to be exactly the same, right? Right? It's not like that into a single mother or other, but that could be exactly the same like that. Now you are the real one, so no doubt about it that you are the person that's doing something wrong in my house. Okay. All right. Appreciate it. Thanks a lot. Okay.
And we use DNA for medical tests such as diagnose the disease in patient and for development of vaccines and PCR. Polymerase chain reaction or PCR is a process to synthesize DNA fragment in a test tube. And if you want to do a PCR test, you should have a PCR component.
Okay, before that, just a minute. Let's see the, have you heard about, maybe 3 years ago? So you are grade 8, right?
5, grade 5.
Grade 5, okay, back 2 years ago.
About 2 years ago. Grade 7, grade 6.
Okay, have you heard about the TB PCR on detecting SARS-CoV-2, the COVID-19?
Oh, yes.
Okay, have you heard about that thing, right? Okay, now think about it. Because of the virus also contains genome. It's a viral genome. Okay, you follow? The virus that infect us also contain genome. It's a genome from virus. Okay, different from our genome, right? Okay, now we want to be sure to make sure that you're infected with the virus, but the virus is so little amount. Think about it, so little to find. Okay, if we have a lot, if we have small amount of the DNA or RNA, whatever, from the virus, what can you do? We know that we have it, but we're not so sure that we have it because the amount of the starting point is so little to see what we have to do. So it means we have the way, even that we have a little amount, but because of that, to make be sure we can amplify the portion of the virus genome. We have more of that to see results, so we can detect them. You clear? You clear? So now we call the basic of that in order to amplify the portion, the segment of DNA we call PCR. Okay, all right guys. Okay, go ahead. Do more.
Okay, so for the reaction, you can see here from the left side is small tubes, and we here like that. So after you amplify you get a lot of DNA inside the small tubes. But the next question is, if we cannot see, how we can detect? So today we're gonna learn about another technique called gel electrophoresis. I will make it easy. This is a method that you can analyze the PCR band inside the gel. So, did you ever eat an agar or jelly before? Just think about that. Today we will make something like that agar, which is called agarose. That will have a small part inside. That when we extract the PCR or we have a PCR products or DNAs, we can just load by the pipette. You remember the pipette last time that you tried? Yes, you use that one to load the samples into the small hole on the top, and then we will run it, and we will stand, and you can see by this technique. So today we will learn about this also. And in here, we also use in our lab. We also use the PCR technique to apply to detect some things that might contaminate into our cell. Next week, you will learn about the cell, right? So in here, the cell that you're gonna learn is supposed to be very clean and no contaminant from any bacteria or something. So we also use the PCR technique to test about mycoplasma test. Next time, maybe we can talk about this later. But today I think we spent a lot of time here, so I think we're gonna go to the lab part, and I will pass it to Beyond, okay?
Okay, go ahead. Let's just ask you one more time. Just to be sure that they understand. Okay. So before you have a fun thing here, I just want to make sure that you understand it. Okay, the products of the PCR, you got the piece of DNA, right? Which is I have you amplified it. Okay? And then you'll be able to see it through running through the gel. Okay? So that's the product right there from the PCR. You follow? It's not the whole genome. It's just a portion of DNA that we detect, that we amplify it out. Okay, that's today you will see it by your own eyes. Can you follow so far? You follow? Okay. All right, go ahead. On มาต่อเลย
Okay, it's a laptop